There we go. All right, so what's kind of nice, I'm like, although you already used to know already, a lot of the stuff we're going to get through at the beginning of today's lecture is a little bit of review of some structures that are in the urinary system, which you already got tested on today, so you should know them already. But our kidneys have a ton of different functions. It's just not to make urine. It helps regulate your total water volume in our body. It regulates total solute concentration that we find in the water. It regulates extracellular fluid ion concentrations. Again, what's an ion? Yep, it has a charge. And I'm like, so it's going to regulate, the amount of sodium, potassium, chloride. It's going to regulate all those ions. It ensures a long-term acid-base balance. Again, can help regulate the pH of our blood. It can remove metabolic wastes. It can remove toxins. It can remove drugs, which is why they test for drugs using urine. It also has endocrine functions, so there's several hormones that the kidneys make. One is a hormone known as renin that helps regulate blood pressure, which we'll get more into next time, a little bit today. Another hormone known as erythropoietin, which should be a little review, because it means making of red blood cells. It's a hormone that comes from the kidneys. And it also helps activate vitamin D, which helps in calcium absorption. So the kidneys have a lot of different functions to them. Lots of diverse functions. So the basic systems, yes, you've got two kidneys. That's what we're going to talk about today in all the major filtering. The ureters, all they do is transport urine from the kidneys down to the bladder. The bladder just stores the urine, and the urethra transports that urine out of the body. I think I took some stuff off on this slide. So if you're like, why am I missing a line? I took it off. Kidney anatomy. Your right kidney is crowded by the liver. Again, so it sits a little bit lower than the left. On top of the kidneys, you have adrenal glands, sometimes called suprarenal. What do you think suprarenal means? And what do you think renal means? Kidney. So the adrenal or suprarenal gland sits on top of the kidneys. Other parts of the kidneys, the ureters, your renal blood vessels, renal artery, renal vein, some lymphatics, and nerves all enter and exit at the hilum, that indent in the kidney. Internal, I was going to say this is like a review from today for lab. The cortex is the outer part. The renal medulla is the inner part that has the columns and the pyramid. The papilla is the tip of each pyramid. This is where all the urine is going to be made and go out of the pyramid into one of those calyces. The renal pelvis, it's a little funnel area right in the middle of that kidney that's going to drain urine out. You've got minor and major calyces that are draining urine from the pyramids. So kind of the overall urine flow, urine's getting made in the pyramids. It then gets out into the minor calyx, which join together to make a major calyx. Goes into the renal pelvis, then goes into the ureter. And then where does it go from there? Bladder. After the ureter, and then after the bladder, the urethra, and then out of the body. Yeah, that wasn't close. A little review, but again, that's kind of lab, so I'm not going to go any of that because you just got tested on it. Blood supply. Yes, the kidneys do filter a whole lot of blood. So since the kidneys cleanse the blood, they have to have obviously lots of blood going to the kidneys. Approximately the renal arteries deliver about a little over a liter, 1,200 milliliters of blood to the kidneys every minute. That's a lot of blood that has to go to kidneys every minute. So over a liter of blood to the kidneys every single minute to get filtered. Arterial flow of blood into and venous flow of blood out of the kidneys follow similar paths. Meaning anywhere you see an artery or an arteriole, you're going to see a vein as well. So you've got arteries and veins running together here. You've got arteries and veins running together in here. We call them cortical veins, cortical arteries. They're all up in the cortex region. So wherever arteries go, veins are right there next to it. The nephrons, these are the things that are actually going to make the urine. You have over 1 million of them in each kidney. Two main parts of the nephron, the renal corpuscle and the renal tubule. The two main parts of the renal corpuscle, you have the glomerulus. This is where you've got all the blood capillaries, highly porous. Now you'll see one of the words, they didn't leave the whiteboard marker. In some of the terms, it's a word that means porous, just in case you're like, I have no idea what this is, because it is a completely new word. If you see the word fenestrated, it means porous, because I know you're going to see it on one of the diagrams as well. So anytime you see fenestrated, it means porous. These capillaries are highly porous, which means we're going to have stuff filtering out of the capillaries. This is the part. We're filtering blood. We're going to make urine. So it allows for filtrate formation. So when we'll start, I'm like, filtrate is not urine yet. And I'm like, we go from blood to filtrate to eventually urine. Filtrate is just the stuff that was filtered. And I'm like, but it's not urine yet. Also, outside of the glomerulus, you have the Bowman's capsule, also known as the glomerular capsule, just a hollow structure that contains that glomerulus. I don't care about a bunch of this. But different parts of the nephron. Now again, I'm like, most of the nephron hangs out in the cortex region. The only main part that's actually hanging down in the medulla part of the kidney are these loop of Henle's and some of the collecting ducts. Now these loops and the collecting ducts, I'm like, that's what's going to give that striped appearance to most of those pyramids. Otherwise, you're going to find that renal corpuscle, the distal and proximal convoluted tubules, all hanging up in the cortex region. But now, inside that glomerulus, or inside the Bowman's capsule where you have the glomerulus, this is what we're talking about. The actual membrane itself in the glomerulus has little holes in it. I don't know how well you can see it, where there's structure, hole, structure, hole, structure. There's holes. So that's the fenestrated endothelium of the glomerulus. There's the porous part, so that you can actually have stuff leaving the blood itself. Now, the rest of it, the renal tubule part, three main parts. You have the proximal convoluted tubule. Again, convoluted means the twisty-turny part. Proximal because it's closest to the renal corpuscle. You have the nephron loop, also called loop of Henle, and the distal convoluted tubule because it's farthest from the renal corpuscle. And then you have, I was going to say we haven't gotten there yet. The proximal convoluted tubule has cuboidal cells. They have dense microvilli. This is where you're going to have brush border cells. Yes, in the intestines they make some of the enzymes. But by having these microvilli, kind of the folds on the folds, you have really large surface area. Because we want really large surface area because it's in this proximal convoluted tubule where you have both reabsorption and secretion taking place. The greater the surface area, the more reabsorption, the more secretion can take place. Now, the proximal convoluted tubule, again, is, yes, stuck up in that cortex region. So here's your proximal convoluted tubule. The nephron loop, it has a descending limb and it has an ascending limb, hence the whole loop part. The proximal descending limb is continuous with this proximal convoluted tubule. It's the exact same width as you start going downward. Then it changes. So this is still part of the loop of Henle or the nephron loop, and it's still the exact same type of tissue on the outside. It's the same as the proximal convoluted. However, then it does start to change. Then you get into that distal descending limb. Now we start having more of a simple squamous epithelium. Why do you think simple squamous? Why? Yeah, for stuff to go across. Again, this is where you have most of your, well, I'm going to say not most of them, like you've got lots of reabsorption and secretion happening up here. However, we have lots of stuff that we're going to still reabsorb out of the loop of Henle or the nephron loop. A lot of this is water, a lot of this is that sodium chloride. So you have to have a thin membrane, and then that thin membrane, as we start to go up and we start getting into that ascending limb, now we're going to change and get to more of either cuboidal or columnar cells. Sometimes it's thin all the way up. Generally it changes to more of a thicker, where you've got cuboidal and columnar cells. So that loop of Henle, that nephron loop, just change in width because there is different tissues found along it. So there is different things being absorbed and secreted in there. Then you have the distal convoluted tubule. You've got some cuboidal cells. There are not as many microvilli. If you don't have as many microvilli, you don't have as much what? You don't have as much surface area. You're not going to have lots of reabsorption. Little stuff might get reabsorbed, but it's not its main job. Most reabsorption happens in the proximal. Some stuff can get secreted here, which means some stuff can get into that actual tubule versus reabsorb it out. It's also confined to the cortex region of the kidney. And the collecting duct, its main job is to receive filtrate from many nephrons. So I was going to say, I'm like, I feel like I should have another picture. So one collecting duct, again, multiple nephrons are going to dump into one same collecting duct. They're showing one. Every single one of these other little things sticking out, that's where another nephron's going to dump in there. They run through the medullary pyramid part, given it the striped appearance, and they do fuse together, and they deliver urine through the papilla, the apex, into the minor calyces, that little funnel at the top part of the pyramid. Now, Mike, so all of that urine is coming from the collecting ducts. You know, Mike, that's just receiving it from all the different nephrons. I was like, it does have some stuff. There is some reabsorption that'll happen out of the collecting duct. We'll get more into that part. We won't get to that, all of that today, what's happening in the collecting duct. Now, there are two types of nephrons. There's one known as a cortical nephron and one called a juxtamedullary nephron. It's a long word. The majority are cortical nephrons. So 85% of all the nephrons are cortical nephrons. Named because they're almost entirely found in the cortex. So all of that proximal, all that distal, and they have a short little nephron loop of Henle. Now, Mike, so the majority are all hanging out in the cortex region, hence the cortical. And then you have the juxtamedullary nephrons, about the other 15%. Their main difference is they have a really long nephron loop, really long loop of Henle. It goes really far down into the medulla, and its main job is to help concentrate urine. So it's going to try to pull as much water out as possible. So this picture looks like your lab. Yeah. And I said, oh, here are nephrons, and we'll learn the difference between them next time. Here we go. So since the majority of this nephron is hanging up in the cortex region, this would be an example of a cortical nephron. This is a nephron that has this huge nephron loop. This would be one of the juxtamedullary nephrons. Now, other than the fact that one has a long nephron loop or one has a long loop of Henle and the other one doesn't, what other big difference do you see? The peritubular capillaries. So the peritubular capillaries are hanging up here surrounding all that proximal, distal, convoluted tubule. Are there any capillaries up there? No, where are they? They're surrounding the nephron loop, or that loop of Henle. Are there any capillaries surrounding the loop of Henle over here? No, and I'm like, so where those capillaries are also different between the two. So one's got a really long nephron loop, mostly in the medullary, in the medulla part of the kidney. The cortical hangs out mostly in the cortex region. Short little nephron loops goes down into the medulla, but the capillaries that surround them are different. Now, well, we already kind of said this. I'm like, so a renal tubule is always associated, every part of the renal tubule is associated with two capillary beds. There's always going to be a glomerulus capillary. And if you are a cortical nephron, you're going to have those peritubular capillaries. If you're a juxtamedullary nephron, you're still going to have a glomerulus. However, you now, instead of having the peritubular capillaries, your capillary is known as this vasa recta, this part that surrounds that nephron loop. So you both still have the glomerulus. One has the vasa recta, and the cortical has the peritubular capillary. Now, why, what's all happening in the capillary beds? The majority, if you're in the glomerulus, filtration. The glomerulus is specialized for filtration. It has all of those pores in there. It's different than other capillary beds. Most capillaries, where we talk about it, we have an artery going in, and we have a vein coming out. You're dropping off oxygen. An arterial takes oxygen, oxygenated blood into a capillary area. We drop off oxygen, and then we've got a vein leaving that capillary. In the glomerulus, leading into it, we have an afferent arteriole, and leaving it, we have the efferent arteriole. They're both arteries. Because is there any dropping off of oxygen here? No, and I'm like, it's different. Most capillaries is for dropping off oxygen. This particular capillary has to do with filtration. And so you have an artery going in, you have an artery coming out. Now, blood pressure that's in that glomerular capillary is really high for two main reasons. One, afferent arterioles are larger in diameter than efferent arterioles. So if you have a really big, or really wide afferent arteriole, and then leaving, you have a more narrow efferent arteriole. What happens to blood pressure when you go from wide area to less area? Yeah, it's going to get stronger. It's kind of when you reduce volume, you increase pressure. And that's what happens here. When I try to take all this blood that's in the afferent arteriole, and I now try to shove it into a narrower efferent arteriole, the blood pressure is higher. So you have a lot of high blood pressure in this glomerulus because of the different sizes in those two arterioles. And the arterioles themselves are a high resistance vessel. That I'm like, they're not going to stretch. They're going to stay as is. So any blood that's in there is going to have a higher pressure. They're not going to expand and give way to have a bigger volume. Now, it's good that we have lots of blood pressure in that glomerulus. And we'll get into that still today. The peritubular capillaries, the other main capillaries, the part that's hanging outside the peritubular or the vasa recta. The peritubular capillaries that are up here, they're a lower pressure. Again, we're coming from the efferent. As we start to branch and go out, we start to become a lower pressure, more porous capillary. And again, anything where you have pores, it's because we have stuff going in or out. So now we can have absorption of water and solutes. So we make filtrate, which now goes into the tubules. However, I may want to reabsorb some of that stuff from the tube back into the blood supply. So the blood supply needs to stay pretty close by. So we can have lots and lots of openings, little areas for stuff to go from a tube into those capillaries. We already kind of did this in lab, but they arise from the efferent arteriole. Blood goes in the afferent, leaves through the efferent, and they're just hanging outside of those renal tubules, only in the cortex region, so you don't see it on that nephron loop. The vasa recta part, if you're a juxtamedullary capillary, or a juxtamedullary nephron, it's long, goes around the entire loop of Henle, it's thin-walled. They still also arise from efferent arterioles, just like on the cortical, except we have them around the nephron loop instead of having peritubular capillaries. Their main job is the formation of concentrated urine. So any water that's going to be in the filtrate inside this tube is going to go from tube into the blood supply. We're going to reabsorb as much water out in that nephron loop that we possibly can. The more water you reabsorb out of that filtrate, the more concentrated your urine is. Now, altogether, we make about 180 liters of filtrate a day. That's a lot of gallons. I was going to say there's about 4-ish liters in a gallon. You do the math. That's a lot of gallons. That's a lot of filtrate that we make every single day. We make about 180 liters of fluid. Only about 1.5 liters actually turns into urine. Again, this is 500 milliliters, so you make about 3 of these worth of urine a day. Again, you drink more water, you're going to make more urine. You drink less water, you'll make less. But approximately, you make about 1.5 liters of urine a day. All because of 3 basic processes, which I'm trying to remember. I know we'll get through glomerular filtration today, and that's probably mostly it. The 3 main processes in urine, you have glomerular filtration, you have tubular reabsorption, and you have tubular secretion. The glomerular filtration, so it's happening inside the glomerulus. We are filtering the blood, And our main job of glomerular filtration is to produce a cell-free, protein-free filtrate. So, Mike, you should not have any cells, you should not have any protein in your urine. Tubular reabsorption, this means about 99% of all the substances that were in the filtrate get reabsorbed back into the blood itself. Again, only about 1% of filtrate actually gets made into urine, which if we go back to the numbers, you make about 180 liters of filtrate a day. You only make about 1.5 liters. There's your 1%. Only about 1% of the filtrate actually gets turned into urine. Everything else, the 99%, all get reabsorbed back. We can use all of it, so we're going to try to reabsorb it all back. And tubular secretion, opposite of tubular reabsorption. Reabsorption is to, get everything back, try to, hold on to and reabsorb everything that we can possibly use over and over and over again. Tubular secretion, we are secreting everything that we don't want in our body to end up in the urine. So we're trying to get rid of it. So we can move substances from blood into the filtrate that will eventually make it into the urine. So kind of the three basic processes. Glomerular filtration, obviously happening up in the glomerulus. This is now where you're making filtrate, which ends up in the renal tubule. Then you can have tubular reabsorption. Any filtrate materials, 99% of it, is going to go from the tube back into a capillary, which luckily right next to the tube you have those peritubular capillaries. Then we have tubular secretion, which means we may have stuff in our blood that we want to get rid of. We can just secrete it into the renal tubule and it ends up in the urine. Again, things like drugs, and I'm like, why they can do drug tests using urine. That's something that, drugs can end up in your blood supply and we can secrete it into the renal tubule and it ends up in the urine. Now, glomerular filtration is a passive process, which means what? Doesn't take any ATP. But we need filtration to happen, so there still has to be some type of energy behind it. The energy that actually causes filtration to happen is hydrostatic pressure. This is the fancy way of saying blood pressure. And we'll talk about different types of blood pressure and different types of pressures coming up. But this is generally your blood pressure. The fact that every time your heart beats, you have pressure in that blood, in those arteries, in the arterioles that are going into the glomerulus. We have a high pressure that's trying to push stuff out of the blood. So it's hydrostatic pressure that's actually pushing different things through the filtration membrane. Again, it's a porous membrane. So between the blood and the glomerulus. So this is where we have blood. Here is our glomerular filtration. Again, you can see the holes. This is that fenestration. These are the pores. Now, Mike, so we're going from blood now into filtrate. Red to yellow. Now, I had it highlighted. I did circle it. Plasma of the blood is getting filtered. I'm like, you shouldn't end up with red blood cells, platelets, white blood cells. Those are too big to actually go through any of these pores. The only thing you're really filtering of the blood is the plasma. What is plasma mostly made up of? It's mostly water, which can easily go through those pores. So water of the plasma can easily go through it, and any solutes that are smaller than plasma proteins. So proteins should not pass. If they're smaller than a plasma protein, they should be able to fit through these pores. And normally no cells pass through. Now, if your kidneys start to become damaged, these pores might start to get wider, and now you end up, or the blood pressure is so high it can shove bigger things through those holes. Then you generally know that, you're destroying your kidney. You would end up having protein in your urine. You might have blood cells in your urine. That usually means your kidney is not working correctly. Now, on occasion, and we can get some molecule stuck in the pores, whether it's a cell, whether it's a big protein. Some large molecules can get stuck in there. There are some glomerular cells that are just like macrophages. They go in and just engulf anything that gets stuck in there. But the filtration membrane itself allows anything smaller than 3 nanometers to pass, which means water can easily go through, glucose can go through, amino acids, which do amino acids make up? They do make up proteins. Proteins are too big to go through, but amino acids are small enough they can go through. Any nitrogenous waste, all can go through. Now, it's good that those proteins do not go through the membrane. There are lots of proteins in your plasma. The biggest protein that's in your plasma, I know we briefly mentioned it when we did blood, is a protein known as albumin. Too big to go through. That's not a bad thing, though. I'm like, what if I want to draw? Again, I'm not a wonderful drawer. There's my Bowman's capsule. Do I have red? And there's my glomerulus, where I'm actually going to have filtration taking place. Now, if proteins would actually go through the membrane and end up in the tubule, what generally happens, water follows solutes. It always, water follows solutes. It's a good key to know. Water follows solutes. A solute is anything that can be dissolved in water. If these proteins could actually fit through, you would have too much water following with it. If you have too much water leaving blood, what's going to happen to your blood? It'll start to become too thick, and you're not going to have enough blood to sustain life. I mean, the majority of your blood is water. And I'm like, you're not going to be able to have enough blood volume circulate around your body anymore to transport oxygen everywhere. So by having plasma proteins stay in the blood, the water will also stay in the blood. Otherwise, there's enough pressure in your blood itself that, in theory, all of the water should be able to go through. That all of that water should get filtered right through. We don't want all the water to end up in the renal tubule. By having some of those proteins stay, it maintains what's known as your colloid osmotic pressure. This colloid osmotic pressure, this is blood pressure due to proteins. That's a weird word. This is blood pressure due to the proteins that are in the blood itself. And I'm like, the fact that there is proteins in there, mostly the albumin, they're holding on to the water itself, larger volume, larger pressure. So it prevents loss of all your water into the space inside of here. Otherwise, all your water would leave your blood. Wouldn't be good. If you have proteins that ended up in here, it usually means there's got some type of membrane problem, damage to the actual kidney itself. It's not wonderful. Sometimes it happens with too high of blood pressure. Now, pressures that affect filtration. So what's actually happening here? Yes, we already talked that there is pressure behind the blood that's going to cause filtration to happen. That's known as the outward pressure. You have one major outward pressure that promotes filtration happening right here. The major pressure that promotes filtration is known as your hydrostatic pressure. So this is your hydrostatic pressure. It's saying, let's make urine. This is blood pressure. It's called your glomerular blood pressure. Blood pressure that's in all those little capillaries. This is the chief force that pushes water and some solutes out of your blood, such as amino acids and sugars. I'm like, it's saying, let's make urine. And it is quite high. Most capillaries have a blood pressure of only about 26 millimeters of mercury. That's a pretty low blood pressure, considering normal blood pressure in your arteries. It's like 120 over 80. Most capillary beds have a blood pressure of around 26, low blood pressure. Glomerular blood pressure is around 55, so it's a higher blood pressure. Again, we're trying to get stuff out of the blood and make it into urine. Again, part of that is because that efferent arteriole, it's a high resistance and it's smaller in diameter than the afferent. However, so one major pressure saying, let's make urine. You, however, to have two other pressures saying, let's not make urine. The one inward pressure is known as the hydrostatic pressure in the capsular space. Inside of here, so in this little space itself, this is my capsular space. Again, here's the Bowman's capsule. there's pressure in there. Just by the fact that there is some filtrate hanging out in here that we just got done making, by having, this isn't an empty space, there is filtrate in here, which means this urine's like, I don't want to go there where there's already filtrate. This is that hydrostatic pressure in the capillary space. So that's one pressure that's saying, okay, slow down, don't make so much urine. The other is the fact there is pressure in the capillaries, or the pull of those proteins. The fact that there's still proteins that don't go through, mainly the albumin, that's saying, okay, we're trying to hold on to the water, we're not going to let it all go through. That's the other pressure that says, let's slow down. Now, your hydrostatic pressure, your main blood pressure, is usually, again, about 55. That's normally 55, saying, let's make urine. The pressure that's in that capsule itself, that's only about 15 millimeters of mercury, and the pressure that's getting pulled from those proteins is about 30. So if we do the math, 55, that's what's saying let's make urine, minus the other two pressures that's saying let's stop, we should have a positive number. Yeah? If this number is positive, urine will be made. As long as the pressure that says, that hydrostatic pressure, as long as that pressure says, let's make urine, and that pressure is bigger than the other two, we can make urine. If your blood pressure drops too far, that affects this number. And and it causes not as much filtration to happen. And it can happen. So, this is your net filtration pressure. Net filtration pressure. Approximately 10 millimeters of mercury. Now, net filtration pressure, what are these three different pressures, is the main factor that determines glomerular filtration rate. How much blood am I filtering in any particular time period? If your blood pressure goes up, what's going to happen to filtration rates? It will also go up. If this goes up, that goes up. And so your filtration rate can change based on that net filtration pressure. So your net filtration rate is just the volume of filtrate that's made every minute by both kidneys. And you make approximately 120, 125 milliliters of filtrate every single minute. That's, 4 minutes and you have this much filtrate. And you think how long we've been here? 4 minutes, you make this much filtrate. That's a lot of filtrate. Now, your filtration rate, again, mostly dependent on blood pressures. Your net filtration pressure. Also, to a minor detail, you're looking at surface area available for filtration to happen. As you start to get older, you start to lose surface area in the capillaries. That starts to go down. And your filtration membrane permeability. How many holes, how many pores are in the capillaries themselves? How big are the holes? That's the filtration membrane permeability. Is it permeable, more permeable in one person than other? Does it change? Oh, I'm stuck. Now, things that regulate the glomerular filtration rate. I know I changed, I think, these next two slides because it was just repeating itself, which was weird. Now, we generally have a pretty constant glomerular filtration rate. We make about 125 milliliters of filtrate every single minute. It allows our kidneys to make lots of filtrate. We can maintain extracellular homeostasis. We maintain the ions. We maintain the fluids. We're fine. We keep everything just fine. We have an intrinsic goal and we have an extrinsic goal for the urinary system. The intrinsic goal, I think inside the kidney, is to maintain glomerular filtration rate of approximately 120, 125 milliliters a minute. We're going to try to maintain that much filtration. Now, we can generally maintain that filtration rate as long as our arterial blood pressure is in that 80 to 180 millimeters of mercury. That's still a big range. Your normal artery blood pressure is around 100, again, 120 over 80. And I'm like, as long as we still keep anywhere in that range, our kidneys are doing just fine. The problem becomes if your blood pressure is too high or too low, we can't filter blood like we should. If this number becomes too high, we start damaging our kidneys. And I'm like, we start, damaging holes. And I'm like, we can start plugging them up with things. And I'm like, we might start getting proteins going through too fast, pulling water out. If it becomes too low, well, if this number goes into the negative, you're not going to make any filtrate. So as long as your arterial blood pressure stays in that range, your kidneys can continue to make 120, 125 milliliters of mercury, or milliliters of filtrate every minute. An extrinsic goal. So that's the intrinsic goal. As our kidney's job is just try to maintain that filtration rate. An extrinsic control. So what our kidneys are trying to do with that filtration rate is to maintain blood pressure. Now, this is why in nursing, I'm like, the urinary system is one of the most important systems, because everything always seems to come back to urinary system. Oh, blood pressure is high, blood pressure is low, kidneys are probably failing. And I'm like, because generally, if your blood pressure changes, your kidneys should be able to try to regulate blood pressure. So an extrinsic control is to maintain your body's blood pressure. If you increase your filtration rate, you make more urine. You're making more filtrate, you're making more urine. Now, if you make more urine, you decrease blood volume. By decreasing blood volume, you decrease blood pressure. So like your kidneys change the blood pressure, and then the vice versa. If you decrease how much urine you're making, you're decreasing urine output. That means you're going to increase blood volume, which increases blood pressure. So like anytime, blood pressure should be regulated by the urinary system. Now, the nervous system and there's, the nervous and endocrine mechanisms also help maintain blood pressure. And I'm like, so if your blood pressure all of a sudden again changes for whatever reason, your nervous system can kick in. You've got endocrine functions, various hormones that we'll talk about that can help kick in. And they might tell, pretty much shut the kidney off that says, we're not going to let you do your job anymore. We're going to take over and regulate blood pressure. So if your blood pressure gets less than 80 or greater than 180, your nervous and endocrine systems start to take over. Otherwise, if your blood pressure is anywhere in that range, your kidneys should be able to maintain blood pressure. They should be able to maintain filtration rate. Now, if for whatever reason your extracellular fluid volume is really low, which means you're going to have a really low blood pressure, low blood volume, low blood pressure, why might you have a really low blood volume? Nope. Think emergency room. Why might you have a low blood volume? You just got done losing a lot of blood. You just got done with surgery and you lost a lot of blood. You were in a car accident, lost a lot of blood. If for whatever reason you all of a sudden have a low blood volume, again, low blood volume, low blood pressure. If you have a low enough blood pressure, are your kidneys going to be able to make urine? Yes or no? No. And I'm like, if that blood pressure goes too low, you're not going to be able to make urine anymore. Your kidneys are going to start to shut down. Again, if you're, severe car accident and you lost a lot of blood, making urine's not really a top priority. Your sympathetic nervous system kicks in. It starts releasing norepinephrine. And epinephrine that's released by the adrenal medulla, based on its name, where do you think you find it? Above the kidneys. And so something above the kidneys helps regulate what the kidneys are doing. So epinephrine is released by the adrenal medulla, right next to the kidneys. It causes systemic vasoconstriction. What does that mean? Constricts the veins. If you constrict the veins and you make them smaller, what does that do to blood pressure? It will increase it. So if your blood pressure is too low, we're going to start trying to increase blood pressure by constricting your blood vessels. It will also constrict the efferent arterial. So the blood that's going in, you're constricting that. If you constrict the blood that's going in, you're not going to have as much you're not going to have as much blood going in, less filtration. Again, if you have low blood volume, making urine is not a top priority. So we're going to decrease that filtration rate. If you don't make as much filtrate, we're not pulling water out to make filtrate, your blood volume goes up. And so blood volume and blood pressure go hand in hand. Like if you've got low blood volume, you have low blood pressure. If you have too much blood volume, you have too high a pressure. Same thing, when you just guzzle a big, huge, 64-ounce thing of water, what does that do to your blood volume? It goes up. If blood volume goes up, blood pressure goes up. If this number goes up, what does filtration have? What happens to filtration? It goes up, and you make more urine. Hence the reason why you drink water, you make more urine. All because you increase blood pressure. Not a ton, but you increase blood pressure. Now there are 3 main hormones that help increase blood pressure. So that's the endocrine system. One's known as renin, another one's known as angiotensin, another one's known as aldosterone. Now I know we're going to get more into aldosterone and a little bit with renin next time. I think they're on the next slide. Really? Write them in. They weren't on the. Is this like the last slide before your review question?
No, there's one more slide, but they're not on there.
What's on the next slide? Yeah, they're right there.
Oh.
They're on the heading.
Oh.
So just write main hormones for raising blood pressure, and then just cross everything else off on the slide. I mean, I could talk about hormones and blood pressure like an entire lecture. Not going to. We'll talk about aldosterone for sure next time, and a little bit with renin. But these are main hormones that increase blood pressure. Again, if your blood pressure is too low because your blood volume is too low, we need to try to get that blood pressure up again. All right, some review. Urine collection occurs in the what part of the kidneys? It is green. In the pelvis parts, that's where everything, it's getting made in the other regions, although it's not even getting made in the columns. And then it collects in the renal pelvis. The blood filtering structure of the kidney is called the what? Is why the glomerulus, that's where blood gets filtered. The urine-forming units of the kidney are the what? These green, the nephrons, make the urine. The vasa recta are associated with which structures in the kidney? It is the juxtamedullary nephrons that have that really long loop of Henle. Which of the following factors contributes to the higher filtration rate in the glomerular capillaries compared to other capillary beds? It's green, all of them. If the fact that you have pores in the capillaries makes them higher filtration rate. And the fact that, yes, you've got that high pressure because of the smaller efferent versus afferent. The extrinsic controls regulate glomerular filtration rate as a means of regulating what? It maintains systemic blood pressure. So the extrinsic control wants to maintain blood pressure. The intrinsic control wants to maintain that glomerular filtration rate.